I'm going to bypass the hair-destroying equipment and try and see if I can just cope with this, okay. So thanks for the invitation to come along to this meeting. I hadn't realized quite what a hoo-ha had been going on in Germany, partly because I was diverted by Brexit, and it has seriously impacted on our lives, as you can imagine, and I think it's going to carry on. So let's enjoy today. Okay. So I've been asked to talk about insights from toxicology, and I think it's fair to say that there's so much work out there that what I've done is I've just picked on some work that's been going on with me in my lab and with my collaborators, and then a couple of other studies, just to illustrate why we think what we're doing is helpful for understanding what's going on. So we've already heard about the London smog of 1952, and we all thought that it was going to be solved by 1956 Clean Air Act, but what happens, as happens throughout life, is that things change. So instead of having the smog-like atmosphere, we have this totally different type of atmosphere that we've seen in the Beijing pictures today and all over the world. And what we're dealing with here is a mixture of gases and particles, but most of my work is on particles, and in particular these very tiny diesel exhaust particles and other smaller particles which can reach the very deep lung. And you'll see in the literature that the incidence of deaths through PM 2.5 has gone up since 1990, and it's the 5th highest ranking mortality risk factor. Now, we heard this morning that GPs and medical practitioners are not so sure about how important that is, because it's a relatively short period of time that you're talking about. But for me, what I think is important is the morbidity. So if we look at those who they're talking about, approximately 50% of the deaths are due to cardiovascular disease, 35% of the deaths that they're measuring here are lung diseases, including asthma, COPD, cystic fibrosis, so it aggravates that. But it's now thought that it might be causal in asthma. So, you know, we don't know. And if people are living with those diseases, then they're uncomfortable. They may not die much earlier, but they're living a very uncomfortable quality of life. And then there's lung cancer. And this is, I think we might have heard this already, but particulate air pollution is now being classified as a carcinogenic agent. Okay, so the respiratory effects are fairly broad. Almost everything is affected. The asthma, COPD, etc. Increased pulmonary infections, particularly in the compromised, the young and the elderly. There's increased symptoms of cough and phlegm, etc., increased lung cancer. But the other thing that we saw on a previous figure, and I'm going to show you again, is that it's affecting the young in terms of lung function and growth and cognition and things like that. So it's not as simple as we originally thought. So I quite like this picture here because what it's showing is a nice healthy lung on the left there, and what they describe as somebody who is affected, who's living in an air pollution particulate environment, and somebody who smokes. And you can see that the lungs are, all three sets of lungs are quite different. I've already talked about these different lung diseases, but now what we're talking about are the cardiovascular effects. for example, changes in heart rate variability and atherosclerosis, and then the systemic effects. It's not working. There we go. Right, let's try this. There we go. So you've seen this figure already, which I stole from the ERS statement in January, not the one that was we heard about. It's not identical to the earlier one. This is a copy of that thing. But so apart from all these pulmonary effects, we're talking about these cardiovascular effects, strokes, cardiac disease, vascular disease, and then every year we're adding something else to all these other labels. It's increasing all the time. And as these particles are delivered to the lung, there's got to be something going on there that's quite important. And this is just to remind you of the differences in the particle sizes and where they deposit in the lung, the coarse, the fine, and the ultrafine. And the cell biology of the lung is so different throughout the respiratory tract that we have to look at each level. My own interest is in what's happening down here in the respiratory units. And so presumably we are getting some bioreactivity in the lung at the gas-liquid interface and in the cells of the lung, but how does this have a knock-on effect in the cardiovasculature and systemically? And why I wanted to show you this is that these are normal lungs, but look at all this is obviously the windpipe here, but look at all the particles and dust that you see collecting in the lungs of these individuals. And it's hard to think that this has no effect on their health. And what I do is I disrupt the lung, me and many others, we disrupt the lung into its component parts, and we study the cells individually. So I've got a nice cell line of this alveolar type 1 cell here that's very thin and sits right next to the blood vessels. And as you can see, this is less than a micron, this micron here, deep. What goes on at this interface? And what do macrophages do? I'm not going to talk about all that today, it's impossible, but those are the sort of things I'm interested in. Okay, and what does toxicity mean? Well, cell death. So unlike what people might think, I don't want to kill all of my cells, but if I do, I'd like to know what mechanisms are going on. Increased oxidative stress, that's a massive factor in the lung. Inflammatory responses. What triggers inflammation? What happens to the immune cells? And then genotoxicity and mutagenicity are very important. Double DNA strand breaks, increased adduct formation that interferes with regeneration of good DNA. So first of all, I just want to talk to you about this study because it was with Jim Zhang and Fan Chung at NHLI and Jim Zhang at Duke. And he generated these particulate matter samples from an engine that was burnt with diesel that had no cerium oxide added, and then added increasing concentrations where one is what you're advised to use, and you use it for economic purposes, you use less petrol, but we decided to look at 0.1 of that and 10 times that, and this is what happens. Particle mass goes down, particle numbers go up, and just to show you that the ceria in these samples also goes up in relation to the amount that you add to the fuel. And when we add these particles at increasing concentrations, and I think we did look at lower concentrations but didn't get an effect, so my students just put the ones that show an effect here. And you can see that you get an increase in this pro-inflammatory mediator interleukin 6. So if we look at the very low concentration of ceria, you can see it has a small effect here, I think no, it doesn't look very different. Sorry, it's this one. It has a small effect when you're looking at the 1:1 ratio, and what it's actually doing is decreasing the effect of the particles. And then when you add 10 times the amount of Syria, you can see that you're actually reversing the effect of the particles themselves. And this is really interesting. It wasn't what we expected. So when we looked at the literature, we discovered that some forms of Syria are antioxidative. And so what this is illustrating or suggesting is that this effect here depends on oxidative stress, and that this effect here is stopping that oxidative stress from occurring. And why are we interested in interleukin-6? For lots of reasons, but this is a very recent paper here that shows that interleukin-6 controls reelin, and reelin regulates pathways induced by, regulates the interaction between neurons during development, and that we know that this can have a knock-on effect and cause autism spectrum disorders in the young. And so this is why looking at something like this, interleukin-6, is important, because interleukin-6 in the lung is dispersed into the system. And then when we looked at oxidative stress visually, and we can measure this quantitatively, you can see that without the Syria, you've got a lot of oxidative stress shown here in red, and as you increase it, you reduce that, supporting the fact that the Syria has an antioxidative effect. So if we look at mutagenicity, in these studies that I'm going to describe, they're interested in the particulate matter as collected and the organic phase that's on the outside of the matter. And they're looking at things like adduct formation and telomere modification. So in this study, where they're using my type 1 cells, shown here in black, and an A549 cell line, which is an adenocarcinoma cell line, they show that this material, this roadside material, background material, coarse and fine, they're showing that the type 1 cells are particularly sensitive to the to one of these samples. And then they went on to show that those samples that were affected, the viability goes down, that there's an increase in production of interleukin-8, which sequesters neutrophils, that there's an increase in DNA damage response, and an increase in sorry, I'm not doing very well on this, in double-stranded DNA damage here. So this is relating the loss of, the increase in cell death to the DNA damage in these cells. I must say that I haven't put any doses on any of these, because when you're a toxicologist, you want to know the mechanisms, and you're not really thinking about the doses so much, but this is 10 micrograms per mil of sample. So if we're talking about telomere length, these are very important. The telomeres protect the chromosomes. Every time the cell divides, the telomeres are shortened, and telomerase lengthens them again, and that protects the rest of the DNA from damage. As we get older, our telomeres become shorter, and we have much more DNA damage. And in this study here, they looked at 2 doses. First of all, 2 doses of particulate material here and here, and they then took the organic phase, washed that off, and looked at it here and here at these concentrations. And you can see that in the blue, the particles, telomerase is reduced in all of the cell samples. But that if you look at just the organic phase, it seems to have just as much of an effect, or if not greater, in terms of telomerase activity, and in the darker bars in terms of telomere length, than the particles themselves. And this suggests that it's what's on the particles rather than the carbon core that is important. But for me, the carbon core is important because that is the vector that is taking this material to the site of action. And then similarly here, when they look at double-stranded DNA damage, they show that the particles themselves are causing double-stranded DNA damage, but that the organic phase is possibly worse. Now, in this study of the type 1 cells in my lab, we were comparing carbon black in the form of Printex, 90 nanometer Printex, with diesel exhaust particles. And we were looking at monolayer integrity using ESIS. And so we put the particles in here, let the cells grow, and we measure over time the ESIS measure here, which is a measure of integrity of the monolayer. And at first, it doesn't look as if there's much difference. But if you look at all those at the bottom of this panel here, the controls up here somewhere, you can see that it's only the diesel exhaust that is actually causing a difference in the monolayer integrity. And when we look at proliferation of the cells, you can see that all of the cells are dividing above the level of control. Here's control. They're all dividing above the I'm sorry, I'm useless at these. You can see that they're all dividing rapidly in order to maintain the monolayer integrity, but that in fact the diesel exhaust exposed cells are dividing even more. Then in this study, this is an in vivo study, which are the other types of studies that we do. The sort of things that they measure are influx of inflammatory cells into the lung. And this is the normal level here. These are 3 bespoke particles here. So we've got diesel exhaust here, we've got concentrated air pollution particles here, and we've got coal fly ash here. And these are particulate matter collected from 2 sites in America. And you can see that although these double up the inflammatory cells, there's big difference, but these mixtures of particles have a much greater effect on the influx of particles into the lung. And this is true for the macrophages, the neutrophils, and the eosinophils. And this shows in the pathology here. This is control. These are the caps, diesel exhaust, and the fly ash there. And then when you look at the 2 particulate matter samples, you can see that there's quite difference in pathology, and you can see that there's an increase in the mucosal area and in the inflammatory cells within the tissue. And this is how they summarized. They measured lots of different things, and they showed that these particulate matter samples induced a whole host of type 2 responses, the sort of response you have in asthma, and inflammatory responses and pathological responses that the other bespoke particles did not induce. They were transient responses. So what we're showing with these studies is increased oxidative stress, inflammation, cell death, increased DNA damage, which would be important in accelerated aging and cancer, abnormal repair and remodeling leading to lung pathology, and also altered physiological responses, as in increased airway hyperreactivity. So I just want to talk to you about some other studies that I've been doing with my colleagues, with Maria Belvisi. She has this lovely cough model in a guinea pig, and they put the particles into the lungs, or they expose the vagus nerve to the particles. And when they exposed the guinea pigs to particles and measured the action potential of the nerves, they showed that DEP activated the nerves. Then when they compared this with isolated nerves from guinea pig and isolated from human, they showed that the same thing happened in human. So this is translational. It's happening not just in guinea pigs, it's happening in human tissue. And then they washed off, they extracted the outer solvents from the outer soluble material from the carbon black, and they looked at the effect of the organic material compared to the particulate material, and then again organic material. And you can see that both the guinea pig and the human react to the organic component, but not to the particles. And so we've done a lot of work on this, and we've been able to show that the activation involves chemosensory nerves, TRPA channel, and not the mechanosensitive nerves. And we had hypothesized that these would be the important ones, not the chemosensitive ones. And we were able to really quite to dissect out exactly what was going on. And it's the polycyclic aromatic hydrocarbons and the aryl hydrocarbon receptor here that is involved, and oxidative stress is involved in terms of activating the TRP channel and triggering the cough response. So in this study, a similar study, but with a very different endpoint, what we did here with Mike Emerson, who's interested in platelet activation. So, okay, I think I've convinced you that there are things going on in the lung, but here what we did was we put the particles into the lungs, and we measured, we injected the mice with platelets that were labeled with indium, and that meant we could measure clotting, platelet aggregation following installation of the particles for 4 hours. And we compared this to carbon black, and we were a bit surprised that carbon black caused quite a lot of neutrophil inflammation. I mean, so did DEP, but carbon black much more so, and mediator production. But then when we looked at platelet aggregation, compared to control, which is the saline light line here, you can see carbon black had no effect on platelet aggregation, whereas the diesel exhaust particles had not only a greater effect, but it took a lot longer to return. So what we're talking about now is the knock-on effect of depositing particles in the lung and that effect on the cardiovasculature. So do the particles get into the lung? This is a study by Mark Miller, and what they looked at was they looked at clinical samples, people who were coming in to have surgery with atherosclerosis, and an animal model of atherosclerosis. And they got the people that were having surgery to inhale gold nanoparticles, and they exposed the animals to gold nanoparticles within the lung. And what they found was that, first of all, you could detect the gold in the atherosclerotic plaques, and that was after just 2 hours of inhalation. They also showed that the gold was excreted by healthy subjects who were exposed during exercise, and they showed that the nanoparticles that they gave to the mice could be located not just in the lung here in red, but also in the atherosclerotic plaques. So the particles are getting out. They are getting out, only a very small percentage get out into the blood. Now, I'm just going to scoot on now. I was going to show you this, but there we go. So for me, the insights from toxicology are that there's a wide range of adverse cellular responses to particles. I think this depends on the cells themselves and the tissues, and also on the individuals and their susceptibility. A lot of things are affected, and I think this varies between individuals: oxidative stress, immune defense, cell senescence, etc., etc. And the susceptibilities and the ability of these populations to respond to these stresses is crucial in their overall response to particulate air pollution. Thank you.